postdoc researcher. So I'm based here in the CCB as part of the Translational Oncology lab. So Michael Brown, who Adam mentioned before, also runs a research lab. So really our job is to try and make these CAR T cells better, come up with new ideas, new ways to test so that hopefully we can get them into the patients. So we all know that brain tumours are particularly challenging tumour to treat. They're often very complex and often the current therapies that we have, including surgery, radiation and chemotherapy, despite these therapies, the tumours will come back. And the survival can vary depending on the tumour type, but it's often very limited. And unfortunately, even though our oncologists and our surgeons do the very best they can, these current treatments that they have to offer can often cause significant side effects which could really impact the quality of life for patients as well. So really our research is trying to develop new therapies which not only are more effective for the brain cancer, but also will hopefully result in less side effects as well for the patients. So just to introduce CAR T cell therapy, which I know Adam's already done briefly, but when, in this therapy we actually take the patient's own blood and we separate particular cells called T cells from the blood. We genetically engineer them within the lab so that they can detect and find the cancer cells. We'll then expand them up and we'll inject them back in the patient, and then hopefully, because they know what they're looking for, they can go find it and kill it. So as Adam mentioned, our CAR T cells are actually already in brain cancer patients. So our lab developed the GD2 targeting CAR T cells. We actually have two different clinical trials running. CARPOS, which is what Adam described, which is run here in Adelaide at the Royal Adelaide Hospital. But we're also using our GD2 CAR T cells to treat children who have DRPG or DNG, and this study is based in the Sydney Children's Hospital. So we actually manufacture the CAR T cells here and send them to Sydney where the children are treated for that. And the early results from these clinical trials so far have shown some tumour shrinkage, which is amazing, and we've also not seen very many treatment-related toxicities so far with the patients that we've treated. However, one thing is that we've found, so this is really good evidence, I guess, that this is a really good strategy for brain cancer and there's some potential there. But unfortunately, we've found that so far the responses that we've seen haven't been long-lasting. So they're working initially, but they don't keep on working. And so really, our job in the lab is to try and come up with new ways to generate the next generation of CAR T cells that are stronger, fitter, and will give better long-term responses. So why don't they work for the long term? So brain tumours, unfortunately, aren't all the same. The brain tumour contains lots of different types of cells. And so while our CAR T cell might be able to eliminate the pink cells really well, the blue cells, if they don't express GD2, our CAR T cells aren't the best at killing those. And so sometimes this will cause the tumour to regrow after the therapy. And so really my research is focusing on trying to overcome this barrier. So one way we're trying to do this is to make our CAR T cells a bit smarter by targeting two different types of proteins on the tumours at the same time. So as we mentioned before, GD2 is one of our targets, so that's the one that's used in the clinic. We've introduced a second target called FAP. So FAP is quite an interesting marker because it's actually expressed on the tumour cells. So hopefully by targeting FAP we can kill those tumour cells that have FAP on them. But it's also expressed on the blood vessels, but specifically the blood vessels within the tumour. So it's not expressed in the normal brain, but specifically on the blood vessel tumour. And so hopefully by targeting two things at the same time, we can get a better response. And I guess the point of targeting the tumour's blood vessel is that's really how it grows. It gets all its nutrients to keep growing. So if we can eliminate that, hopefully we can create a stronger and more coordinated response. So here's a bit of science for you. So this is how we take this idea, we make these CAR T cells and how we test them in the lab. So we can take some glioblastoma cells and we can put a coloured fluorescent protein on their surface. So in this scenario, I've taken some glioblastoma cells and I've made them red if they've got GD2, and I've made them green if they've got FAP. I mix them together in a dish and I treat them with the CAR T cells, and at the end of the study I can ask, well, which cells did they kill, the green ones or the red ones or both? So if we look at some data, if we just treat these cells with T cells that don't know what they're looking for, you can see that I've still got red and green cells left at the end of the study. When I treat them just with GD2 CAR T cells, you can see they do a really good job at getting rid of the red cells, but they've left those green FAP-expressing cells behind. When we treat this combination with our dual CAR T cells, we aim both, we can completely eliminate the tumour cells from that dish. So this is a really good way for us to test if the CAR T cells are doing what we think they're doing in the lab. So from here, we're really trying to optimise these CAR T cells, putting them through lots of different tests to make sure they do exactly what we think they're going to do and that they're going to work long term. Once we're happy with that, we then test them in some carefully designed mouse models. So this allows us to look at what happens in a tumour. So a tumour is very different to some cells growing in a dish, so that's really a good test for them. From them, we then refine our approach based on what we see in those studies, and then hopefully we can use this to generate some data to move our new CAR T cells towards clinical trials. So we're really grateful for the grant funding that we've received from the NRF, because this is really helping us to accelerate progress towards creating more durable CAR T cells that are stronger, fitter, and able to do a better job. And this is actually the first in-field strategy that we know of that's targeting the tumour blood vessels, not just the tumour. And I don't do this alone, so as part of this grant, I've got a team that work with me. So Abby's doing a lot of this work as part of her PhD project. We also collaborate with Veronica, who is based at Women's and Children's Hospital, and she's got some great expertise that she's bringing into this project. And we're also supported by Lisa, who's here today, who brings in a lot of expertise on targeting FAP. And as I said, I'm part of quite a large research team, as you can see here, that's led by Professor Michael Brown, and we're all really together trying to work to make these CAR T cells even stronger and better to hopefully improve the outcomes for patients in the future. All right. Yeah.
We've got time for a couple of questions for Nicole. What people are thinking, I guess, and maybe this is a tough question, but I guess the trials that are happening at the moment are relatively small trials with only a small number of patients, because this is to actually generate the CAR T cells is quite a process to go through. So if there's successful outcomes, how do you then translate that into a larger scale trial, and what sort of infrastructure do you need, and is that likely to be driven out of Adelaide?
Yes, I mean that would be the hope. I mean, the fact that we've managed to get our facility up and running, we've got the expertise. I guess as we go through these projects, we improve the way we're doing it. We come up with new ideas that might make it cheaper, more feasible along the way. We've got different ways to test to make sure that our end product is the right product to go forward and to produce the right data to push that. I would like to say yes, that we could get it up in Adelaide, but it would take a lot of
I think it takes
support.
I guess it takes a lot of support. It takes a lot of money, and the idea is really to have CAR T manufactured close to where the cutting edge research is happening. Yes. I think is the key thing, right?
Yeah. So while we are manufacturing and shipping them to Sydney, the location is a big factor and complicates it slightly. So building our capacity in Adelaide would certainly build our capacity to deliver this to more patients. Sure.
Any other questions?
How long does it take to manufacture sufficient cells
for the clinical trial?
Yeah.
Yeah. So at the moment we collect the T cells from the patients. It takes us about 2 weeks in the lab to grow them up to the amount that we need to infuse into a patient. And then it takes us about 2 weeks to perform all of the quality testing that we need to make sure that they're safe to infuse into the patient. So from the time of blood collection to the time they're ready to infuse, it's about 4 weeks.
So the ones that you manufacture, the original cells that you're expanding on, are taken from patients with a tumour.
Yes.
So is there any chance that they'd be rejected by a recipient that you're transferring them to, the excess ones?
So in this study, the patient's own T cells are the T cells that we will inject back into the patient. So that's precisely right to prevent rejection, because T cells are one of those cells that they only like to be in their own, they'll be recognised as foreign by anyone else. There is a lot of work to try and develop off-the-shelf immunotherapies. T cells probably aren't the key for that, but there are some other types of blood cells that we can use this technology which won't necessarily be recognised as foreign. So yeah, at the moment we're limited in that we can only use the T cells from the patient themselves. And sometimes the patient's T cells might not be that healthy because of prior treatments and stuff that they've had. So we're doing a bit of work in the lab to look at that and make sure we understand it. But at the moment, T cells are just for their own patients. But in the future, I think there's potential that we could have off-the-shelf options that could be given to other patients.
Thank you.
No worries.